Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami, microbiologist. Today in this video lecture we are going to see another culture media, that is a Deoxycholate Citrate agar medium. Now this is the culture media that is specially used for the isolation of Salmonella as well as Shigella species. And this media is actually modified a little bit by using Leifson formula. Now then we have the pH of this medium, so the pH will be 7.3 to 7.5. that is the inhibitory for gram-positive bacteria if we maintain the pH. Now citrate salt concentration in this formula are really important because it will inhibit the gram-positive as well as other intestinal normal flora bacteria. Okay, so this is a little bit modification in this media, that's why it's really important. Now this media is selective as well as differential because we mostly use it for the enteric pathogens. Now this is the culture media, as you can see, deoxycholate without any growth, so it will be look like in this form. Now let's see the composition, what type of the ingredient are present in this media. So this media contain lactose, HI solid, proteose, peptone. So all these three things are 10 gram respectively. Sodium deoxycholate 5.0 gram, neutral red 0.025 gram, sodium citrate 20 gram, ferric ammonium citrate 2 gram, agar 30 gram, and distilled water 1 liter. So all these are the ingredient in 1 liter. Now let's see the principle of this media. So what are the function of HI solid? So HI solid actually providing the carbon and nitrogen source. So when these two sources are available, so what happen? There will be no growth of the coliform bacteria. Next we have proteose peptone that will provide carbon, nitrogen, vitamin and mineral which are necessary for the bacterial growth. We have coliform bacteria as well as gram-positive bacteria that are inhibited or it is suppressed, means the growth of these bacteria are suppressed due to what? Due to the presence of sodium deoxycholate citrate, and we have ferric ammonium citrate. Then we have dipotassium buffer which is actually the buffer means maintain the pH. We have lactose that is help to differentiate enteric bacilli, because we better note that there are enteric bacilli that are lactose fermenter, and due to this way it will produce red colony, and there are some enteric bacilli that are non-lactose fermented, so it will produce colorless colonies. So it is due to the fermentation and non-fermentation of the lactose. Then we have the lactose degradation if occurs. So what happen? It will change the medium pH into acidic, and due to this way what we get colonies. So these colonies are means colorless colonies, and it will indicate the neutral red into new, means it will turn the neutral red its color to a red indicator. These colonies usually are also surrounded by a turbid zone precipitated deoxycholic acid due to the acidification of the medium. We have sodium deoxycholate that will combine with the neutral red in acidic environment, and due to this way it will cause the solution precipitation of the deoxycholate. Then we have the reduction of ferric ammonium citrate. Due to the reduction of ferric ammonium citrate to iron sulfate, what will happen? We will get blackish colonies, or we can say blackish iron sulfide formation. So Salmonella and Shigella, it will not ferment the lactose, but we have Salmonella that will produce H2S. So when H2S is produced, what will happen? Colorless colony will form with black center. So that's how the principle of this media, and it is how working, and we will get the colony of the bacteria. Now then we have the preparation of this media. We will prepare it by taking 70.52 gram of the media into 1000 ml distilled water, and then we will heat to boil to dissolve the media completely. Remember, again in this case we will not autoclave it, because it will destroy the selectivity of the media. Then we have to cool down it till to 45 to 50 degrees Celsius. After that we will pour it into the plates, and once the media is dry, so then we will use it. Now we will take the specimen like feces, rectal swab. So from these specimen we will get the bacterial colony, and we will inoculate the specimen into the media, and after 24-hour inoculation we will get the bacterial colony. This is how the media is prepared, and the culture is performed, and the media is used. Then we have the result on this media. So we have although we cannot use it for the E. coli, but if let's suppose you see the E. coli, so there will be a poor growth, or it will show pink colonies with bile precipitate negative reaction for H2S, means there is no production of H2S in this case. Salmonella enteritis typhorium aboni, these type of the bacteria showing very good and luxuriant growth, because this media is specially for these types of bacteria. And we get colorless positive reaction for the production of H2S black center colonies. We have Shigella flexneri, it also showing good growth, colorless colony. Shigella sonia also showing colonies, and their colonies are smooth, initially colorless. Moreover, it is pale pink on further incubation if we perform, and due to late lactose fermentation. This is how the Shigella sonia will be look like. We have Enterobacter Klebsiella, it will show large pale mucoid colony with pink center. Then we have Enterococci, which are not growing, sometimes less growing. We have Yersinia enterocolitica, it will also showing colorless colonies. And we have Proteus profidentia and Pseudomonas. I forget to write the name of the Pseudomonas here, but we have also Pseudomonas. It will showing colorless, often glossy with large central black dot and fishy color. Fishy odor will be in the case of Proteus and profidentia, but in case of Pseudomonas we get pungent or like smell. Now these are the growth of the bacteria on this culture media. As you can see, these black colonies are the growth of Salmonella enteritis typhorium and aboni. If you want to find out any one of the bacteria from this colony, how will you differentiate either it is Salmonella enteritis typhorium or aboni? So for that you will perform the different biochemical test, so then you will be make sure that what type of bacteria of Salmonella is it. These are the growth of Shigella sonia. As you can see, colorless colony. These are the mucoid colony of Enterobacter and Klebsiella. A little bit difference between Klebsiella and Enterobacter is that Enterobacter's colony is a little bit small as compared to the Klebsiella. Then we have Shigella flexneria and Salmonella paratyphi. Both have the same colony, but if you want to differentiate it further, so then you will perform the biochemical test further. Then these are the growth of the Pseudomonas and Proteus. The difference is just that Proteus will show fish-like odor, Pseudomonas will show pungent smells. Then we have Yersinia enterocolitica. This is the colorless colony, as you can see. So these are the bacterial colony using these media. Then we have the uses of this media. As we talked earlier, means that we use this media for the isolation and differentiation of gram-negative enteric bacilli in the laboratory setting. Moreover, we also using this type of media for in case of dysentery to isolate Salmonella strain that is causing food poisoning and Salmonella paratyphi organisms. Let's see the limitation of this culture media. Sometime what happen on DCA media, mean deoxycholate media, what will happen? We have Salmonella growth, but there is a lot of Salmonella growth. Sometimes Salmonella enteritis, Salmonella, we have Salmonella Shigella, we have Salmonella typhorium. Every colonies are black center colonies. So if you want it to clarify it, so then you have to perform other biochemical tests, and you will differentiate the salmonella from each other using another media which we call that XLD media, which I already explained. You can go back to my channel and watch this video. Then we have the now another most important point is that as we talked that we cannot autoclave it, so we have to heat boil it. Now after the heat boiling, what we will do, we have to a little bit cool down it, so then we will pour it. But if we not doing like that and we put the cholate after addition this, so what will happen, the media will be soft and it will be very difficult for us to handle. So that's why it's really necessary to and we can say it is heat sensitive, so we have to be very careful. Then we have third point. Now the third point says that if let's suppose you have the stool and urine specimen and you want to examine it, so if you are using DCA media, means deoxycholate media, so with this media you must use McConkey and bismuth sulfate agar medium as well, so you will not just dependent on this media. Okay, now the next point says that this media is actually this media do not have a lot of nutrition, so sometimes there will be a less growth of other bacteria as well. And if you want the fifth point and the last point says that if you want to confirm the bacteria 100%, then what will you do, you have to perform different biochemical test. So this is all about the deoxycholate agar medium. I hope this video is helpful for you. Thank you so much for watching.